Good afternoon. Today's class is anatomy of leaf, the internal structure of leaf. Today we will discuss. Let me share. Sharing option లేదు రిక్వెస్ట్ అడిగిన రిక్వెస్ట్ పంపినా డిక్లైన్ చేశారు. The TS of leaf shows three parts. The first part is epidermal layers. Yeah, have a look. So this is today's class is TS of dicot leaf in the beginning. Later we'll discuss the TS of monocot leaf also. The dicot leaf is also known as dorsiventral leaf, since the upper surface and lower surface of the dicot leaf, externally they show the differences. They differ externally and internally also they have the difference. The external appearance of the adaxial surface, that is upper epidermis, and the lower epidermis, subaxial side, are different. They show some slight difference. That's why the dorsal side is upper side, ventral side is lower side. They have the difference. So that's why this dicot leaf is also called as dorsiventral leaf. Dorsiventral leaf. Here, so on the slide, the image shows the upper epidermis. This is upper epidermis, and the lower side also, the same epidermis will be there. Let us go for detailed discussion. So this is under the microscope, the transverse section of epidermis appears like this. This is the dorsiventral leaf. This is the dorsal side. The upper epidermis is called as dorsal side. This is the lower epidermis. This is the lower epidermis. This lower epidermis is called ventral side. I'll repeat once again. Upper epidermis is called dorsal and the lower epidermis is called ventral. Both the epidermal layers are single layered of cells. They are made up of a single layer of cells. Same like root and stem, you can observe the epidermis is made up of a single layer of cells. Single layer of cells. So this is upper epidermis. The lower epidermis is also made up of single layer of cells. The part of the leaf present between the both the epidermal layers, that is upper epidermis and lower epidermis, this portion is called ground tissue of the leaf. I'll repeat once again. The tissue present between both the epidermal layers is called ground tissue of the leaf. That ground tissue of the leaf is named as mesophyll. Meso means middle, phyll means leaf. Between the two epidermal layers, a simple chlorenchymatous tissue will be seen. The tissue is termed as mesophyll. In Greek, meso means middle, phyll means leaf. This is the middle portion of the leaf. So this is mesophyll. This is the second part. And the third part of leaf is the circular central portion is seen, which is surrounded by a dark pink color tissue. In diagram, it is seen as a dark pink color sheet, that is a spherical structure which is seen at the center of the mesophyll is called vascular bundle. This is vascular bundle. Here, at the midrib region, even at the lateral rib region also, lateral vein region also, you can find vascular bundles. But the midrib region, there is a large single vascular bundle could be seen. This is the vascular bundle. So I'll repeat once again. The TS of leaf shows epidermis, that is the first part. The second one is mesophyll. The third one is vascular bundle. Epidermis is the uppermost and lowermost layer on both the sides, on adaxial side and abaxial side. A single layer of cells are seen. This is called epidermis. Already we have studied about the epidermis characters in roots and stem. The same cells are seen here also. The cells are barrel shaped, rectangular in shape, closely packed. They are closely packed without any intercellular spaces. Generally, they are colorless. Since they do not have chloroplast. Since leaf is the aerial part, which is a flat structure, this is the leaf lamina TS. This is a flat structure. It needs protection. That's why the externally on both the sides, epidermis is covered with thin cuticle. Yesterday in stem structure also, we have seen cuticle is a thin dead layer made up of waxy substance. Cutin is a wax-like substance. It protects leaf from external forces. Like it protects the leaf from adverse environmental conditions, and also it checks the loss of water, that is called transpiration. It checks transpiration. The entire epidermis gives protection to the inner parts. So slight difference is there in lower epidermis. You can see here and there aerating pores, minute microscopic aerating pores are seen. These are minute microscopic aerating pores. They are nothing but they are stomata. Even in upper epidermis also, a few stomata may be also present. But on the lower side, the number of stomata are more. What do you mean by stomata? Stomata are minute microscopic aerating pores, which are generally found on leaves. And in young stems also, you can find such aerating pores. Those pores are named as stomata. In upper epidermis, the number of stomata are very less. In lower epidermis, the number of stomata are comparatively more. If 1 and 2 stomata may be there, here 3-4 stomata also will be seen. But at the midrib region, there is no stomata here. This is the midrib region, which is swollen parts. This midrib region has no stomata. No stomata. So the stomata helps in respiration, transpiration. That means exchanges of gases. Generally, at the time of photosynthesis, plants get carbon dioxide from the atmosphere through these pores only. Carbon dioxide enters into the plant body through the stomata. And the oxygen which is released during photosynthesis will be released through the stomata into the surroundings, into the atmosphere. That means exchanges of gases takes place through these pores. They help in exchanges of gases. That is particularly during photosynthesis and even in respiration. But in respiration, the exchange of gases is reverse. They breathe in oxygen like any animal. Plants also need oxygen, carbon dioxide. When there is no photosynthesis, plants get oxygen from the atmosphere and they release, like us, they also release carbon dioxide in the night. They release carbon dioxide in the night. They get oxygen from the atmosphere. That is during respiration. Respiration takes place throughout the day and night. But the photosynthesis takes place only during daytime. That's why during daytime, plants breathe in carbon dioxide. They take up carbon dioxide through these stomata and they breathe out, they release oxygen. But in respiration, this is reverse. Like us, they take up oxygen, they get oxygen from the atmosphere and they release carbon dioxide during respiration. That means what is the main role of stomata? They help in exchange of gases. They help in exchanges of gases. And also, you know, plants get water through the root system with the help of root hairs. Root hairs are unicellular external outgrowths. They are very delicate part of the root system. They absorb water and minerals from the soil. That water is to be translocated through the xylem and reach each and every part of the plant body. Generally, 98% of water is lost in the form of vapor through the aerial pores, that is through the stomata. The loss of water in the form of vapor through the stomata in the presence of sunlight is called transpiration. Transpiration is nothing but loss of water. Transpiration is necessary for the plants. It helps in ascent of sap. But they get 100% water. Out of 100%, 98% of water is lost by this process. That's why transpiration is also called as necessary evil. Necessary evil. It is a necessary process. If transpiration is not there, the ascent of sap may not be occurring. That's why for the ascent of sap, for the transpiration pull, for the movement of water through the xylem from root system to shoot system, the transpiration is very important in tall trees. That's why transpiration is also called as necessary evil. Why it is called necessary evil? It is necessary for the ascent of sap. Why it is evil? Because plants are losing 98% of water through the process. And the stomata are helping them. Stomata exchanges gases and also they lose water in the form of vapor. This is about epidermis. And here in dicot leaf, you can find epidermal hairs. Multicellular epidermal hairs are also there. In upper epidermis also, epidermal hairs will be there. In lower epidermis also, epidermal hairs will be there. They are absent in some leaves like nerium, mango. These epidermal hairs are absent. But in tridox, sunflower, in most of the plants, these epidermal hairs are present. They are called trichomes. Trichomes are present in most of the dicot plants. But in some plants, which are leathery in texture like nerium and mango, these trichomes are absent. Here you can see multicellular trichomes are there, but they are made up of uniseriate cells. These trichomes are called uniseriate trichomes or epidermal hairs. Epidermal hairs also help in checking the transpiration. When there is a deficit, deficit that is water content is less in the soil, they check the transpiration. They check the transpiration. The stomata are closed during night time. That's why during night time, if there is no sunlight, they close the stomata. So stomata are open during daytime and closed during night time. That means the rate of photosynthesis is there, the rate of transpiration is there during daytime is maximum. But at night time, the rate of respiration is also very less and there is no light phase in the and dark phase also even absent in the night time. That's why stomata are closed during night time. For this, later slides, we can see the structure of stomata. These are the parts. You can see the cuticle. This is upper epidermis, and this is mesophyll. These are the both the epidermal layers. Let us see the dicot leaves are dorsiventral. Why they are called dorsiventral? Since dorsal side is upper side, ventral side is lower side. They are different structurally and physically. They are different, even though they appear similar, but slight anatomical differences are there. Both the surfaces are covered with thin cuticle, but there are few or no stomata on upper epidermis, and many stomata are present in the lower epidermis. Upper epidermis is also called as dorsal side, or lower epidermis is called ventral side or ventral epidermis. And the other name is also there. Upper epidermis is also known as adaxial side. And the lower epidermis is called abaxial. This is the typical dicot leaf. And already we have discussed about the names. Epidermis, mesophyll, and vascular bundles. Generally in dicot leaf, there is a large central vascular bundle could be seen at the midrib region. This is the midrib region. This is the vascular bundle. Okay, let us move to the next slide. See, epidermis is the outermost tissue. Generally, cells are barrel shaped. Cells are barrel shaped, composed of thin layer cells. All the cells are epidermis like stem and root. They are living cells. And the outer side, you can see the cuticle. That is waxy layer. It is made up of cutin. Cutin is a waxy substance. This epidermis plays a very important role in exchanges of gases. As earlier we have discussed, it has a tiny pores. Let us see. So this is stomata. When you have a close look under the microscope, this pinpoint portion, if it is enlarged, it is magnified part of the leaf. Under the microscope, you can find such structures, such aerating pores. These are stomata. See, in the schematic diagram, you can see the carbon dioxide is moving in and water and oxygen are moving out through the stomata. Stomata is enclosed with a special modified epidermal cells. Generally, epidermal cells are barrel shaped, rectangular in shape. But in stomata, see, stomata is surrounded by two modified epidermal cells which are bean shaped or kidney shaped. These bean shaped or kidney shaped cells are called guard cells since they are guarding stoma. Stoma is the opening of the stomata. And the guard cells contain chloroplast. These are the kidney shaped or bean shaped cells are guard cells. They contain chloroplast. The guard cells help in, since they are guarding stoma, opening of the stomatal complex, they are called guard cells. Guard cells are modified epidermal cells. They are in different shape. Under the microscope, in dicot leaf, you can find bean-shaped or kidney-shaped structures in epidermis. Around the stoma, there are two modified epidermal cells could be seen. These modified epidermal cells, which are in different shapes, called God cells, since they are guarding stoma. And also, they help in movement of stomata. Opening and closing of stomata is taking place with the help of these modified epidermal cells. These modified epidermal cells help in opening and closing of stomata. They are modified epidermal cells. They contain chloroplasts. They are in different size. They enclose opening, microscopic, minute opening called stoma. Stoma is the opening. These are God cells. And adjacent cells, which are next to the God cells, are called accessory cells. These are accessory cells. These accessory cells help in movement of stomata to the God cells. This is about stomata. The more number of stomata are there on lower side, that is abaxial side or ventral side of the leaf. Ventral side is lower side. Lower side is also called as ventral side or abaxial side. On lower side, you can find many number of stomata comparatively. On upper epidermis, few stomata are there. The stomata help in exchanges of gases and loss of water. This loss of water is called transpiration. This is about epidermis. On both the sides, epidermis is covered with a thin layer, a waxy coating called cuticle layer. Cuticle layer is made up of cutin. Cutin. This is about epidermis, first part of leaf. Generally, sunflower leaf is a typical example for dipart. Okay. See the second part, some mesophil. So in the beginning of this session, already I told you, so meso means middle. In Greek, meso means middle. Phil means leaf. This is the middle part of the leaf. And this is also called as ground tissue. Ground tissue of the leaf is called mesophyll. The tissue present between the two epidermal layers. I'll repeat once again. Mesophyll is the ground tissue of the leaf present between the two epidermal layers. Between upper epidermis and lower epidermis, this ground chlorenchymatous green color parenchyma is called chlorenchymatous tissue that is present between the two layers. Generally, in dicot leaf, you can see towards the just below the upper epidermis, towards the upper side, towards the ventral side, the mesophyll is having a vertically elongated, closely packed cells with large number of chloroplasts. See this portion. Generally, it is 2 to 3 layered structure. This tissue is called palisade tissue. This is called palisade. Palisade tissue is present just below the upper epidermis. Towards the ventral side of the mesophyll, this vertically elongated, closely packed cells with narrow or small intercellular spaces. These are the small white color structures could be seen here. Between the green dark structures are there. The each dark vertically elongated is called palisade chlorenchyma. This palisade chlorenchyma is nothing but it is a palisade part of palisade tissue. This is the mesophyll part lies just below the upper epidermis. That's why if you see the color of the coloration is also different. As I told you, this leaf is called dorsiventral leaf. This dicot leaf is called dorsiventral leaf. Why it is called dorsiventral leaf? Since the dorsal side and ventral sides are different externally and internally. This is the internal difference. See, towards the lower epidermis, you can see loosely packed with large cellular spaces or air spaces could be seen. This tissue is called spongy tissue or spongy parenchyma or spongy parenchyma. The towards upper epidermis, mesophyll is made up of palisade tissue, and towards lower epidermis, spongy tissue are there. That means the mesophyll is differentiated into palisade and spongy tissues in dicot leaf. That's why the dicot leaf is named as dorsiventral leaf. The externally, the always upper side of the leaf is dark in color. dark green in color. Dark green in color. Why it is dark green in color? Since the palisade parenchyma cells are closely packed and with large number of chloroplasts. Since the large number of chloroplasts, it imparts the number of chloroplasts impart the color to the cell. That's why they are dark green in color. That's why if you observe the upper side of the dicot leaf, it is dark green in color. And the lower side, there is a loosely packed, simple green color parenchyma tissue is seen. This is called spongy tissue. It is having large air spaces. Yeah, these large air spaces are clearly visible near the stomata. Here, this is stomata. This is also stomata. This is also stomata. So near the stomata, the air spaces are very clearly seen. They help in exchanges of gases. And the spongy cells or spongy parenchymatous cells are having the shape is also different. They are having less number of chloroplasts. That's why this lower part is light green in color. Dorsal side, that is upper side of the leaf, is always dark color, dark green in color. And the lower side is light green in color. Since the internally palisade tissue is located just below the upper epidermis and cells are closely packed. Cells are vertically elongated, columnar cells. They are like columns with small or narrow intercellular spaces. One or two layered cell tissue is seen just below the upper epidermis. That is palisade parenchyma. This is spongy parenchyma. Spongy parenchyma is made up of oval size or circular shaped, small, loosely packed cells with large intercellular spaces could be seen here. So this is spongy tissue. This is about mesophyll. What is the function of mesophyll? Photosynthesis. Photosynthesis. And in photosynthesis, glucose is being prepared or is synthesized as an end product. That glucose is also reserved in the mesophyll cells for temporarily. So this is about mesophyll. And the next one is vascular bundle. Vascular bundle. Let us see. Let us have a close look. See, this is vascular bundle. The central vascular bundle is found in dicot leaf where there is a vein, mid vein or mid rib. This is the rib portion. Just above the rib portion or vascular bundle region, you can see a loosely packed simple parenchymatous tissue. Loosely packed simple parenchymatous tissue is seen. This is a conjunctive tissue. This is a simple conjunctive tissue. And here there is a sheath around vascular bundle. There is a sheath around vascular bundle. This sheath is called bundle sheath. The bundle sheath is parenchymatous. Bundle sheath is parenchymatous. So here the tissue, the ground tissue present near the midrib, surrounding the vascular bundle. This tissue is not differentiated into palisade and spongy parenchyma. It is a simple parenchyma. Generally, it is colorless. Very few chloroplasts are there. That's why at the midrib region, externally, we can see the light green color. Light green color, generally achlorophyllous. So this is simple parenchyma. And this is this pink color sheath-like structure, which is surrounding the vascular bundle, is called bundle sheath. Bundle sheath is parenchymatous. Here, if you see, if you have a close look at the vascular bundle, here see, this is the xylem. These are the xylem elements, xylem vessels. These are xylem vessels. This is phloem. Light blue color part is phloem, and this is xylem. There are sclerites or xylem vessels are there. These are xylem vessels. They bring water and minerals from the root. They bring water and minerals from the root. They help in translocation of water or transportation of water. In leaf, dicot leaf, xylem is present towards the upper epidermis. Xylem is present towards upper epidermis. This is upper epidermis. This is xylem. And the lower epidermis, in each vascular bundle, you can find a patch of phloem. Phloem helps in translocation of organic solutes. The food materials which are being produced, which are synthesized during photosynthesis by the mesophyll cells, that is palisade parenchyma and spongy parenchyma, prepares food materials by the process called photosynthesis. The end product of photosynthesis is glucose. That glucose is being transported to the different parts of the plant body through the phloem. This is the phloem tissue. In phloem, you can see sieve tubes and sieve tubes. Phloem fibers are absent. In xylem, xylem fibers are absent. There are no fibers. Xylem fibers are absent in xylem. Phloem fibers are absent in phloem. Only here xylem vessels are there and few xylem elements, the other xylem elements may be also seen in the xylem tissue of the vascular bundle. Whereas in phloem tissue of the vascular bundle in leaf, the phloem fibers are absent. Phloem fibers are absent, only sieve elements. So that is sieve tubes are present. So this is about vascular bundle. And if you see, if you observe the xylem and phloem, there is no strip of cambium like stem. That's why they are side by side. That's why they are called, these VBs are called conjoint vascular bundle. And they are on the same radius. That's why they are collateral vascular bundle. And there is no strip of vascular cambium, that is vascular meristem is absent. Meristematic patch is absent here. That's why the vascular bundle in leaf is called closed. Which type of vascular bundles are present in leaf? Conjoint, collateral, and closed type of vascular bundles are present in leaf. This is vascular bundle. This is about dicot leaf. This is also important for essay. If you get this as essay, you need to draw the diagram. You have to write, you have to label the diagram. Then you have to write, you have to describe in detail under the three different subheadings. So this is about dorsiventral leaf anatomy or dicot leaf anatomy. So next, this is monocot leaf. So monocot best example is grass. That is particularly maize. Maize is taken as an example for monocot leaf. Here in monocot, you can see the similar type of mesophyll is there. This is not differentiated into palisade and spongy. Palisade and spongy tissues are not there. The similar type of ground tissue is there between the two epidermal layers. That's why monocot leaf is also called as isobilateral leaf. I'll repeat once again. Monocot leaf is also called as isobilateral leaf. Iso means same, similar. Bi means both. Both the sides, lateral, bilateral, both the sides, upper side and lower side. The epidermal layers are same. The mesophyll is same. Number of stomata also same here. See. And the cuticle is thick here. You can see the thickness of cuticle layer. Comparatively, in dicot leaf, a thin cuticle was there, a waxy layer that is made of cutin. Thin membrane was there in dicots. But in monocots like grasses and maize in sugarcane, you can find a thick cuticle. Thick cuticle could be seen. So this is about epidermis. One important character you need to notice, just so you have to remember that point. That is, on both the epidermal layers, you can find the stomata. They are equal in number. They are equally distributed. On both upper epidermis and lower epidermis, you can find same number of stomata. Not like dicots. In dicots, the number of stomata are less on upper side and more in lower side. But here in this case, on dorsal and ventral side, the number of stomata are same. Not different. They are same. That's why it is called isobilateral. But there is a slight difference. Externally, the epidermal layer is made up of thick cuticle. That point you need to remember. And one more thing you have to, I think you have noticed in the slide, see the image. Some of the cells in upper epidermis are large in size. Generally, they are large in size. They store water. They are transparent. They store water and they are transparent. Such large epidermal cells are seen in monocot leaf. They are named as motor cells or bulliform cells. They help in rolling and unrolling of the leaf. When the conditions are adverse, leaf rolls into a small structure. Then the conditions become favorable and it unrolls. It becomes normal. Unrolling and unrolling of leaf blade, these bulliform cells are helpful. Motor cells are helpful since they store water. Since they store water, they are large in size. When the conditions are adverse, the bulliform cells or motor cells lose water, they shrink, they become smaller. That's why the leaf rolls on its own axis, on its midrib part. And when the conditions become normal, these bulliform cells or motor cells get water from the surrounding cells and it unrolls. They become large in size. That's why these bulliform cells help in rolling and unrolling of leaf based on the conditions of surroundings. So this is here, bulliform cells are absent here. There are no motor cells. Such large cells are not there in lower epidermis. That is the difference you need to remember. And here, trichomes are absent. That is a noticeable change. Here, there are no epidermal hairs. In some plants like xerophytes, you can find, like in sugarcane, you can find a sharp trichomes are seen. Sharp pointed trichomes could be seen in sugarcane. In maize also, a thick cuticle is there. In almost all the grasses, thick cuticle is there. Presence of thick cuticle is xerophytic nature. You know the xerophyte? Xerophytes are desert plants. They grow in waterless conditions. They can adverse conditions, desert conditions. Such plants are called xerophytes. Even they grow in the soils where there is a less water content. Even though the water is very less, they can survive. Such plants are called xerophytes. The presence of thick cuticle on the surface of xerophytic character. Xerophytic character. Grass plants, almost all the grass plants show xerophytic character. This is about epidermis. Next, mesophyll. Mesophyll is the part of the ground leaf, which is simple, a green color parenchyma. Like dicot, in dicot, palisade and spongy tissues were there, but in monocot, a similar type of cells are there. It is same on both upper epidermis side and lower epidermis side. The mesophyll is similar cells. It is having a similar cells, and with few intercellular spaces. Some of the intercellular spaces are smaller and some are larger. Generally, these large air spaces could be seen near the substomatal region. This is the substomatal region. Where there is a stomata, it has to store air. It helps in exchanges of gases. The number of chloroplast in cells are same in all the cells. Throughout the mesophyll, all the cells are having the same number of chloroplasts. Same number of chloroplasts are there. See here. So one important thing you can, I think, already have noticed. In dicot leaf, there was only a single vascular bundle was there. Here, different sizes of vascular bundles could be seen here. And one vascular bundle where there is a midrib, you can find a large, bigger or larger vascular bundle could be seen here. Here they are smaller vascular bundles. Since, you know, the venation is parallel venation. Many veins runs parallelly throughout the leaf lamina. Since the monocot leaf is having parallel venation, they have many vascular bundles are arranged. Could be seen in the anatomy of the monocot leaf. At the center of the mesophyll, many vascular bundles are seen. Since there is a parallel venation is seen in monocots. In almost all the grasses, the venation is parallel venation. So the way this is at the vein region, you can find vascular bundle. This is the midrib region. There is a prominent rib vein will be there in each leaf. That rib is called, that vein is called midrib. And these are lateral veins. Lateral veins. And these veins are divided into veinlets. They are parallel. They are parallel to each other. That's why you can find more number of vascular bundles in mesophyll. Here, one more difference you can see. So there is a sclerenchymatous patch in the midrib region. It is a connective tissue, a simple supporting tissue is seen. In dicot, it was parenchyma, but in monocot, it is sclerenchyma. It is a dead tissue. It provides mechanical support. The presence of sclerenchymatous bundle sheath is the character of xerophytic plants. That's why the grass plants show the xerophytic nature. The presence of sclerenchymatous bundle sheath. These are bundle sheath extensions. These are called bundle sheath extensions. They provide mechanical support to the veins or midribs. So this is about mesophyll and vascular bundle. If we have a close look, each vascular bundle is similar to that of dicot vascular bundle also. But the number of xylem vessels are few here, very few. Only two metaxylems are seen, which are large in size, and protoxylem is there. A single protoxylem is there. In some plants, you can find protoxylem lacuna, lysogenous cavity like monocot stem. Also could be seen in the vascular bundles of some monocot plants. It's very few number of phloem is similar to that of dicot leaf. But the bundle sheath extensions in dicot leaf, parenchymatous. In monocot leaf, sclerenchymatous. In monocot leaf, it is sclerenchymatous. And each vascular bundle is conjoint, collateral, closed, same like dicot leaf. So let us see. So just now I told you, and mesophyll is not differentiated. These are the identical features of monocot leaf. Both the epidermal layers both bear stomata, but the slight difference is this. Structurally, the epidermis, upper epidermis is structurally different. Some of the cells are larger in size. They are called motor cells or bulliform cells. They are present in upper epidermis. A mesophyll is not differentiated into palisade and spongy tissue. Just now we have seen the bundle sheath extensions are sclerenchymatous. These are bundle sheath extensions. They are sclerenchymatous. And these bulliform cells help in rolling of the leaf lamina. And one important point you need to remember, the upper epidermal cells contain silica granules. Silica granules. That's why the cells are very sharp. The leaves of sugarcane, leaves are very sharp. They cut the skin if you go bare body. The leaves cut. The leaves are very sharp because of the presence of silica granules in the epidermal cells. Some of the epidermal cells. That's why they are very sharp. Your skin will, you will get scratches if you walk in the sugarcane field with bare body. That scratches because of the silica. Silica granules will be there in some of the epidermal cells. So have a look, comparative study. So the upper part TS of leaf is monocot leaf. The lower one is dicot leaf. Now we can compare. See, the upper epidermis is also having the stomata, lower epidermis is also having stomata in monocot leaf. Here in the figure shows two stomata, here also two stomata. Here you can see very few stomata. So here in the figure you cannot see the stomata on upper epidermis. Here you can see on lower epidermis you can find two stomata. Here bulliforms are there, but in the figure it is not shown. In upper epidermis here, and both the epidermal cells are same. Here thick cuticle will be there, here thin cuticle will be there. In dicot leaf, cuticle is thin. In monocot leaf, cuticle layer is thick. And the mesophyll is differentiated here. See, in dicot leaf, mesophyll is differentiated into palisade. This is palisade. Dark color columnar cells are there. Here a single layer of cells were there. This is spongy tissue. Spongy tissue is having large air spaces. These are the large air spaces. This is palisade. This is spongy. Here the mesophyll is same. That's why the monocot leaf is isobilateral leaf. Dicot leaf is dorsiventral leaf. This is the dorsal side. This is the ventral side. Here you can see vascular bundles. Here a single large vascular bundle is present at the midrib region. But here you can see at the midrib region, this is a prominent vein. You can see a large vascular bundle. But in monocot, there is a parallel venation is there. That's why the small vascular bundles are there. These are the veins of parallel, vascular bundles of parallel veins. Parallel veins. And each vascular bundle, you can see very few xylem vessels are there. Here, a series of xylem vessels are there. Many xylem vessels are there here. Dicot root, stem, leaf. In dicot leaf, xylem vessels are more. In monocot leaf, xylem vessels are very less. And one more prominent difference is there. These are bundle sheath extension. This is a simple ground tissue, parenchymatous tissue surrounding the midrib region is simple parenchyma. Here it is sclerenchyma patches are there. These are bundle sheath extensions. Here also bundle sheath is there. This is simple parenchyma. Here also bundle sheath is there. But this is sclerenchyma bundle sheath. This is parenchyma bundle sheath. So how can you identify the monocot leaf? Presence of upper and lower epidermal layers. Mesophyll is not differentiated. Each vascular bundle is surrounded by sclerenchymatous bundle sheath, and many vascular bundles are arranged parallelly in the mesophyll. Absence of cambium. That's why the vascular bundles are called closed. And vascular bundles are collateral and closed type. Stomata on both the sides, equal in number. Trichomes are absent. Thick cuticle. Presence of motor cells or bulliform cells in the upper epidermis. Earlier I said almost all the grass plants show the xerophytic characters in the leaves. Thick cuticle on both the epidermal layers. Presence of motor cells. When the conditions are adverse, leaf roll back. That bulliform cells helps. Or motor cells helps in rolling and unrolling of leaf lamina. So that is the special feature of that is xerophytic character. And sclerenchymatous bundle sheath extensions and bundle sheath. Stomata in furrows. Furrows means deep notches will be there. In that notches, they are present. And one important point I need to clarify. There in dicot leaf, stomata are there. That is surrounded by two God cells. God cells are kidney shaped or bean shaped in dicot leaf. Whereas here, dumbbell shaped stomata, God cells are there in monocot leaves. What is the shape of God cells in monocot leaf? Dumbbell shaped God cells. In dicot leaf, kidney shaped or bean shaped leaf. So God cells. So this is overview. So this is the description. Monocots leaf here. Silicate deposition is there in monocots. There is no silicate deposition. In upper epidermis, bulliform cells are there. Here, there are no motor cells or bulliform cells in dicot leaf. Mesophyll is not differentiated in monocot. Here it is differentiated into palisade and spongy tissue. And vascular bundles, here in monocot, sclerenchymatous bundle sheath extensions and bundle sheath. Here parenchymatous bundle sheath. So next, Symmetry, here isobilateral, because both upper and lower side similar, same structure. Here dorsiventral, upper side and lower side are distinguished separately. Even the color is also different. The upper side in dicot leaf is dark green in color, lower side light green in color. The number of stomata are equally distributed on both the surfaces in monocots, but in dicots unevenly distributed. Upper side very few number of stomata and the lower side more number of stomata. That condition is called hypostomatous. Hypo means below. And the lower layer, here amphistomatous means on both the layers. On both the layers you can find stomata. That condition is regarded as called as amphistomatous condition. Here hypostomatous condition. Here God cells in monocot are dumbbell shaped. Here God cells in dicot are kidney shaped. So this is about internal structure of leaf, anatomy of leaf. On both the leaves we have finished. So tomorrow we have a class that is secondary growth in dicot stems. So all the best. Let me close the session. Thank you.